Day before the exam, or 2 days before the exam, depending on how you want to look at it, right? So I did get the review session posted finally, and it's on the schedule page, and you can watch that if you wish. And as I said, I'm going to be gone, so I'll be traveling tomorrow. I'll probably be hard to reach tomorrow, at least during the day, if you want to get a hold of me. So I will be available probably tomorrow evening sometime. In any event, if you have any questions, let me know. I've written the exam, and I can tell you briefly about the exam. It's obviously the same format as you saw last time. It's got it's got 3 parts. First part, same as before. That corresponds to 16 points. The second part of the exam is short answer, as before, and that corresponds to 42 points. And the third part of the exam is longer answer, and that corresponds to 42 points also. So the total of that is 100 points. There is an extra credit question on there because you guys have been singing loudly, so that keeps those extra credit questions going. I know there's a lot of information, as I've talked to several of you. What I would encourage you to think about is try to see the big picture. Okay? Try to see the big picture as you're going through and studying material. That's the number one encouragement I would give to you about the exam. As I said, Dr. Merrill will be giving the exam in here on Friday, so please line up like I've asked you to do in the past, starting at the end with number 1, 3, 5, 7, 9, etc. So you're sitting in every other seat, and then, of course, whatever other instructions he may give you, please follow through with him. Okay? All right, so yesterday I talked about fatty acid oxidation, and I want to finish up the oxidation of unsaturated fatty acids today. And then we'll talk about the synthesis of fatty acids and see how far we get with that. This topic of lipid metabolism is the only one in the course that I actually scheduled 3 days for. So it tells you that there's a lot of different considerations in lipid metabolism, and we really only scratched the surface of lipid metabolism. So a lot of things to think about here. Well, what I described to you yesterday was a process of oxidation of fatty acids that occurs on saturated fatty acids, meaning all single bonds. Okay? Now, what's happening in the cell, of course, is that cells have unsaturated fatty acids that must also be used, because if we decided we weren't going to oxidize our unsaturated fatty acids, we would be losing an awful lot of energy that's stored in those unsaturated fatty acids. So cells have ways of oxidizing unsaturated fatty acids. And I'm going to try to keep this as simple as I can, although, to be honest, it is a fairly complex topic. Okay. Let's see. Where do I want to start? Let's say I have a fatty acid like what you see here, oleic acid linked to coenzyme A, otherwise known as enoyl-CoA. All right. So oleic acid has has 18 carbons. It has a double bond at position 9. So when the oxidation of this fatty acid starts, remember all the actions between carbons 2 and 3, and carbons 2 and 3 don't really enter the picture if the double bond is out at position 9. At least they don't enter the picture to start. So beta oxidation starts and clips off some of those 2 carbon pieces until, in this case, we go through 3 cycles, and we get to the circumstance that you see here. All right? So this originally had 9 carbons, all right, before we got to that carbon here. We clip off 6 of those, and we're left with 1, 2, 3. So there is the double bond now in position between carbons number 3 and 4. All right? Well, we want that double bond to be between position 2 and 3. Okay? And a double bond being between position 3 and 4 won't work. The enzyme won't handle it, won't touch it. All right? Not only won't it work because it's between the wrong 2 carbons, the bond is in the wrong configuration. Okay? Remember when I showed you yesterday that between positions 2 and 3, we had a trans bond. Most fatty acids that we get in a cell have cis bonds. Okay, we talked about trans fats and trans fatty acids before. All right? So when they're floating around the cell, whether they're as part of fats or they're part of membranes or whatever they're part of, the natural fatty acids that aren't being oxidized have cis double bonds. So if we're going to get this fatty acid ready for oxidation, we have to not only change the position of where the double bond is, we also have to change the structure of the double bond. Okay? Well, you're going to like this next enzyme because it makes it all very simple. All right? The next enzyme is called enoyl-CoA isomerase, and that's probably a reasonable name to know. Okay? Enoyl-CoA isomerase, right, does a very simple thing. It takes a bond that's between carbons 3 and 4 in the cis configuration, and it converts it into a bond that's between carbons 2 and 3 in the trans configuration. Exactly what you need. How convenient. Right? Very, very convenient. And that's, in fact, now in a configuration that it's just exactly as if it were a part of beta oxidation. All right? If we had gone through the dehydrogenation reaction, that's what we would have, would be this guy right here. The next step after dehydrogenation would be addition of water, which is what you see here. We have the hydroxyl. We would next have the oxidation of that hydroxyl to a ketone, and we would have thiolytic cleavage. So by doing this one simple step with this one simple enzyme, we have made a substrate that will go through the rest of beta oxidation just fine. Okay? So that's not complicated. That's simple, right? You want to see the complicated? No, don't show me the complicated. But I must show you the complicated. No, don't show me the complicated. But I must show you the complicated. Okay? Oh, man, when it starts getting hard to read, you know it's going to get bad, right? All right. I'm going to keep this relatively simple. All right. Here is a polyunsaturated fatty acid, and they don't like to cooperate like the monounsaturated fatty acid that you just saw. Okay? And that's because these guys are spaced in an inconvenient manner. All right? So what happens with this polyunsaturated fatty acid? Okay? Well, we go through beta oxidation as before until we get down to a substrate that the cell can handle. And look at here. We've got 3,4, and we've got a cis double bond. Well, you know how to handle a 3,4 with a cis double bond. That's very simple. All you have to do is add enoyl-CoA isomerase, and it converts a 3,4 cis into a 2,3 trans. No brainer. That's exactly what it does. And so we add water, we oxidize, okay, and we clip off a 2-carbon piece. Okay? We clip off a 2-carbon piece. Now we start beta oxidation again. We pull off the hydrogens in the dehydrogenation reaction. We get the trans double bond there, but look over here next door, we've got a cis double bond between carbons number 1, 2, 3, 4, and 5. Well, here we've got 2 double bonds. One's in the cis, one's in the trans, and the enzyme is getting pretty unhappy. We would like to add water across this and do that, but the enzyme that adds water won't touch that guy. Okay? So it's time to do something else. The something else that it does is with an enzyme that's got a mouthful of a name that I'm not going to ask you to memorize. And it's doing 2 things. First, it's converting 2 double bonds into 1 double bond. What would it take to do that? Is that an oxidation or a reduction? Converting 2 double bonds into 1 double bond. That is a reduction, which means we need electrons. And here is one of the places where we see NADPH being involved. NADPH is donating the electrons. So we're converting 2 double bonds into 1 double bond. And what it's making is something that you can relate to. A cis between carbons 3 and 4. Well, you know how to deal with a cis between carbons 3 and 4. You add our friend enoyl-CoA isomerase, and you've got a trans between 2 and 3, and you go merrily along your way. Now, that wasn't too bad. All right? Am I going to ask you to memorize these structures? No. Okay. Do you need to understand what's happening here? Sure. Okay. Polyunsaturated fatty acids are going to have different considerations than monounsaturated fatty acids. Okay. There's one thing that you should know. Okay. Polyunsaturated fatty acids will get to a substrate that has two double bonds that are next to each other that the enzyme is not happy with. This requires a second enzyme to convert two double bonds into one. When it's done that, enoyl-CoA isomerase takes over and does its thing. That's the upshot of what's happening with the oxidation of this polyunsaturated fatty acid. Now, that's a simplification over what actually happens in nature, but I think simplification is good. It's the big picture that matters here, not the exact details about what's happening. Everybody with me? Yawns? Yep. Okay. Any questions? Everybody's thinking about the exam. That's what's happening, I can tell. All right. The last thing I want to say with respect to breakdown of lipids is a very important consideration. And it's a very important consideration because of the fact that we can't do the glyoxylate cycle. Remember in the glyoxylate cycle, turn around, each time you go around the circle, you generate an extra oxaloacetate, and that extra oxaloacetate can be converted into glucose through gluconeogenesis. And our body needs glucose. Our brain is dependent upon the rest of our body for glucose because our brain can't make glucose. It gets it delivered to it by the bloodstream. If we run out of glucose, okay, we have problems. You know, people who are hypoglycemic, they will faint. They faint because they're not getting enough glucose in their brain to keep them awake. Okay? That's why they faint. Well, if that's severe enough, okay, and you don't have a backup source of energy, you can die. Okay? Well, fortunately, we have a backup source of energy that will usually kick in. Okay? And the backup source of energy that we have is something called ketone bodies. Ketone bodies allow us animals who don't have a glyoxylate cycle to get energy from fatty acids. So our brain has a backup, okay? The backup is it's going to get some energy from fatty acids that are stored elsewhere in my body, all right? And so now I need to tell you how that happens, all right? Well, what's the end product of beta oxidation? The end product of beta oxidation is acetyl-CoA. So I have plenty of acetyl-CoA when I start breaking down fat. Lots of acetyl-CoA. If I have no glucose, then instead of oxidizing that acetyl-CoA in the citric acid cycle in my liver is going to say, whoa, got to get some energy to the brain. And so it starts packaging those acetyl-CoAs up into making ketone bodies. So acetyl-CoA is used to make ketone bodies. Right? How does it start? It starts with two acetyl-CoAs. And we put those two acetyl-CoAs together with an enzyme I talked about yesterday, thiolase. Wow, thiolase is doing both of those things? Well, it's not all that complicated. If you take the thiolase reaction upwards, that was the last step in the oxidation of a fatty acid yesterday. It took that 4-carbon piece and broke it into two 2-carbon pieces. In this case, it's taking two 2-carbon pieces and putting them into a 4-carbon piece. It's just the reverse of the reaction you saw yesterday at the very end of beta oxidation. So it's just simply reversing a reaction. All right, well, two acetyl-CoAs going together make a 4-carbon piece. Okay? And there's actually another acetyl-CoA that comes in to make a 6-carbon piece, and there's some rearrangement that happens. And that 6-carbon piece turns out to be very, very important later. Okay? The 6-carbon piece that's there is called beta-hydroxy-beta-methyl-glutaryl-CoA, which you're much more likely to call HMG-CoA. We'll talk a lot more about HMG-CoA later because HMG-CoA is produced not only in ketone body synthesis, but it's also produced in cholesterol synthesis. Okay? So what we see here are two pathways that are overlapping. The pathway to make ketone bodies overlaps partly with the pathway to make cholesterol. We'll talk about cholesterol later. All right? At this point, they diverge. Okay? Ketone bodies go one direction that you're going to see here. Cholesterol goes a different direction. We're not going to talk about yet. Okay? So we're going to follow ketone body synthesis. Well, at this point, we have 6 carbons in there. As I said, there's a little bit of a rearrangement. The CoA pulls away one of the 2 carbon pieces, and we're left with a 4 carbon piece. The 4 carbon piece has the name acetoacetate. Okay? Acetoacetate is a ketone body. We have just made a ketone body. That ketone body can go into our bloodstream. It's soluble in our plasma. It will travel to our brain. Okay? And when it gets to our brain, the reverse of the process I just described to you will happen. That is, we'll go from here, up to here, up to here, and finally, we'll end up with 2 acetyl-CoAs. Our brain just got delivered energy because our brain can run the citric acid cycle using those 2 acetyl-CoAs. So ketone bodies are delivering acetyl-CoAs to the brain to keep the brain alive. Everybody understand that? If we could make glucose from acetyl-CoA, we would do that, but we can't make glucose from acetyl-CoA, so we make ketone bodies to feed the brain, other parts of the body that need that energy. Questions? This is as dead as I've seen the class all year. Yes, sir? Yep. You know, that's a very interesting question. It's not related to that phosphorylation, no. But yeah, it's a very interesting question because acetyl-CoA actually is needed to synthesize cholesterol. Okay? And so the more acetyl-CoA we can deliver to the brain, the more cholesterol that the brain can actually make. Okay? So that could be a consideration in making brain cholesterol. I had never thought about that before, but that's certainly a consideration, yes. Okay? Not affecting the regulation, but affecting the substrate. Yes? Low glucose will cause this cycle to be favored. That's correct. Oxaloacetate's not involved in making ketones. No, not in making ketone bodies. Okay. Low amounts of oxaloacetate would typically be handled by the cell breaking down aspartic acid and making oxaloacetate. So as long as there were aspartic acid available, which there usually would be, if you didn't have sufficient aspartic acid to do that, what would happen is your body would start breaking down your muscles. That's what happens in starvation, for example. One of the reasons that muscles atrophy is the body is using amino acids to make intermediates necessary to keep the rest of the body alive. Okay? It's possible to use amino acids to make glucose, so I don't leave you the impression we don't have other ways of making glucose. We do. And as the body needs to make glucose, one of the things it will do is it'll break down muscle to get the intermediates necessary to make the minimal amount of glucose that it needs. Yes? How many days without eating? Boy, I can't tell you the answer to that question. It's going to depend on the person to some extent. To give you an idea in terms of glucose, your body basically has about 1 day supply of glucose that it keeps on it. Okay? So if you were to stop eating for, let's say, a couple of days, you would really start, pulling down your glucose. You say, well, 1 day would do it. Well, yeah, but remember that you can make glucose from, amino acids and so forth. So it would take a while to really get down low. But after a couple days, you would start seeing this sort of thing happening. That would be my opinion. Yeah. 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 Everybody's fascinated with ketosis for some reason. Okay. I think that there probably are some benefits to doing that. But like everything else that we see that goes on with fads that people do, fads are almost always taken to excess. And ketosis is one of those things that I think is being taken to excess, to be honest with you. So I'm not sure I remember what your question was, but that's a concern that I have. What's that? Yeah. I can't give you I can't tell you that the answer to that. Sorry. Yeah. I'm also supposed to, the legal authorities tell me I'm officially supposed to say that I don't give medical advice either. So I can't give you medical advice because I'm not a physician. The class before us had medical doctors that perhaps could do that, right? So, okay. Other thoughts about this? Well, what's happening down here? That's one thing I haven't said, the very bottom, all right? What's those other things that are happening there? Well, it turns out that acetoacetate is actually fairly unstable in the body. Okay? So as it's traveling through the bloodstream, what happens is it will spontaneously fall apart. And when it spontaneously falls apart, it will lose a carboxyl group, this one right here on the very end. It will lose that carboxyl group and make carbon dioxide. The other product that it makes is acetone. Right? Now, acetone on a person's breath can be either A, they're really low in glucose and they're trying to get there, or B, it can mean they've got a severe health problem that they're not aware of. So if you smell acetone on somebody's breath, and it does happen, if you smell acetone on somebody's breath, you should talk to them and let them know that. Because some people discover that they're diabetic by just that mechanism I just described to you. They don't know they're diabetic, and somebody smells acetone on their breath. Diabetics have problems with glucose, not glucose metabolism, but glucose delivery and levels. So diabetics will go high in glucose, and then they will go very low in glucose. And when they go very low in glucose, their body's making ketone bodies and making acetone. So again, if you ever smell acetone on somebody's breath, you should make them aware of it and perhaps suggest they see a physician because that's an indication of problems. Okay. Well, then what about this guy over here? Well, this guy over here, beta-hydroxybutyrate, is a protection against acetoacetate. All this is a reduction to convert this ketone into an alcohol, and that actually helps to stabilize the molecule. So people refer to beta-hydroxybutyrate as another ketone body, although technically it's not even a ketone. But it can be readily converted back into a ketone body. So beta-hydroxybutyrate is a more stable way of delivering this in the body. Okay, so ketone bodies, important for energy. All right, we've talked about breakdown. Let's talk about making fatty acids. And the good news I have for you here is that the making of fatty acids is chemically very similar to the oxidation of fatty acids. Very similar. There's really one additional step, and that's basically it. Okay? Fatty acid oxidation occurs in the matrix of the mitochondrion. Fatty acid synthesis occurs in the cytoplasm. The starting materials for the synthesis of fatty acids is acetyl-CoA. Okay? That's what you need to make fatty acids. When do cells move acetyl-CoA out into the cytoplasm? Well, I've already described the situation once, but I'll remind you of what it is. All right? Whenever citrate starts to accumulate in the mitochondrion, it means that the citric acid cycle is not running. Citrate will accumulate. And when citrate accumulates, the mitochondrion says, oh, time to kick it out. And so the mitochondrion will kick out citrate as it accumulates. And when it kicks it out, citrate gets broken down into acetyl-CoA and oxaloacetate. This is how acetyl-CoA is moved out of the mitochondrion. Okay? Now I'm going to give you one more insight. Okay? Look at what's happened in the body. The body has taken pyruvate, which came from glycolysis, and it moved it into the mitochondrion where it made acetyl-CoA. And here the citric acid cycle was plugged up, so it moved acetyl-CoA out here. All right? What does this mean? It means the following. It means that the more I push the synthesis of pyruvate and the less exercise I do, the more I'm going to see this process that you see on the screen happen. That is, high carbohydrate diets are going to favor high levels of pyruvate because that's what glycolysis does. High levels of pyruvate with no exercise, because in no exercise, I'm not running the citric acid cycle. I kick acetyl-CoA back out into the cytoplasm so I can make fatty acids. You want to worry about an obesity epidemic? You just saw it. High levels of carbs are going to do that. Okay? There's no question that high levels of carbs are going to favor the synthesis of fatty acids and favor the synthesis of fat. Okay? Message, reduce your carb intake. Number one. Number two, go jogging with me in the mornings. Okay? Because that is going, you say, look at him, right? But that is going to reduce, all right, your substrates for making fatty acids. Okay. Well, we've got to make fatty acids now, all right? And by the way, this oxaloacetate, what happens to it? It gets moved right back into the mitochondria. It moves right back in very easily, and consequently, it just keeps delivering and delivering and delivering all that stuff. Okay. How are we going to synthesize it? Well, it turns out that the cell has to do a little two-step to start the process. You've seen a two-step before. When we went through gluconeogenesis, you saw that we started with pyruvate, and we added a carbon dioxide and made oxaloacetate, which then we took the carbon dioxide off and we made PEP, right? You guys remember that? No? Yes? You should remember because that's coming up on your exam. That's the biggest question on the exam, right? So maybe, maybe not. I'm joking. Who knows? All right. In any event, the point is that in making fatty acids, we have to do a two-step as well. The starting material is acetyl-CoA. There's an enzyme whose name you definitely should know called I got pyruvate on the head. Called acetyl-CoA carboxylase. Acetyl-CoA carboxylase. This enzyme is the most important enzyme for controlling the synthesis of fatty acids. Acetyl-CoA carboxylase. It's the only enzyme in the synthesis of fatty acids that's regulated. Okay? Its regulation is interesting, and I'll tell you about that in a little bit. But it catalyzes the reaction that you see on the screen. Here's the first of our two-step. We're putting a carbon dioxide, in this case a bicarbonate, on there. We're converting a 2-carbon acetyl-CoA into a 3-carbon malonyl-CoA. All right? That's one of the starting materials for making a fatty acid. Okay? Everybody with me? That energy took ATP. That reaction took ATP. Okay? And it took ATP energy. And we made a 3-carbon intermediate. All right. There's the mechanism of the reaction, which I'm not going to hold you responsible for. But I will tell you that there's the coenzyme biotin, which is an essential nutrient in our diet. Biotin is important for carboxylation type reactions I've talked about before. Okay? So here you saw a carboxylation reaction when we went from pyruvate to oxaloacetate. Here's another carboxylation reaction going from acetyl-CoA to malonyl-CoA. And again, biotin's involved. All right? We're not going to worry about the mechanism. All right? I'm going to come back and talk about the regulation of the enzyme in a minute. Okay? But instead, we're first going to go ahead and synthesize a fatty acid. So we've made one of the important intermediates, malonyl-CoA. Right? Cells don't always do things in logical ways, as I've commented to you before. Right? We make malonyl-CoA, and then the cell says, oh, we don't want that CoA on there. Okay? So it takes the CoA off, and it replaces it with something called acyl carrier protein, or ACP. You can call it ACP. So the CoA is replaced by ACP. So now I have malonyl-ACP. I had malonyl-CoA, now I've got malonyl-ACP. All right? Everybody with me? Okay. We don't have to worry about anything except the fact we're replacing CoA with ACP. All right? So we end up with malonyl-ACP right here. We can do the same thing with acetyl-CoA. So acetyl-CoA can be replaced, the CoA can be replaced by an ACP. That's important because acetyl-ACP turns out to be one of the substrates as well. All right? So we're getting ready to make a fatty acid. We have to have two things to start. One thing we have to start is acetyl-ACP. The other thing we have to start is malonyl-ACP. Both of these are necessary to start the synthesis process. Okay? I'll let you digest that for a minute. Okay? The next step is joining everybody together. So we're going to join the malonyl-ACP to the acetyl-ACP. That would add 3 carbons to 2 carbons. And if we didn't do anything else, we would have 5 carbons linked to ACP. Well, just like the 2-step that we did in gluconeogenesis, where we lost that carboxyl to make PEP, we're going to lose a carboxyl to make a 4-carbon intermediate. So we put the carbon dioxide on, we pull the carbon dioxide off. The difference now is we have a 4-carbon molecule linked to ACP. I don't care that you know the name. Okay? If somebody asked me the name without it being on the screen, I probably wouldn't remember it myself. Okay? If I can't remember it, you guys don't have to remember it. That's one of my rules. Okay? But what I do have, I know. All right? It looks just like that ketone body we just saw. There's the carbonyl end, and there's the ketone. Everything else now is the reverse of what we saw in fatty acid beta oxidation. Okay? Everything else is the reverse. In fatty acid beta oxidation, we convert a hydroxyl to a ketone. In fatty acid synthesis, we're going to convert a ketone to a hydroxyl. That is a reduction, and look, NADPH. Now I have a hydroxyl. Okay? In beta oxidation, I add water across a double bond to put a hydroxyl on. In fatty acid synthesis, I pull water out to make the double bond. And there's my double bond. Okay? Notice it's in the trans configuration. In fatty acid oxidation, to make the double bond, I dehydrogenate a saturated fatty acid. In fatty acid synthesis, I add hydrogen across the double bond, and now I've got a fatty acid that's saturated. Okay? If we go from down to up, that would be oxidation. You've already seen this. Look, dehydrogenation, addition of water, oxidation to a ketone. That's what happened in fatty acid oxidation. We're simply reversing that process. Now, the good news I have for you is, my God, look at those enzyme names, those enzyme names, those enzyme names. You're going to have to know the enzyme name. What's the good news, right? The good news is these are all part of one enzyme, and that's the name you need to know. The name of the enzyme that incorporates all of these activities is a very complicated name. It's called fatty acid synthase. synthase. Fatty acid synthase is a very interesting enzyme. It incorporates many activities, and it actually works like a clock. What you see here is this guy being rotated. Here's the first reaction, the second reaction, the third reaction, the fourth reaction. Repeat the cycle over and over and over. Okay? This enzyme actually has a circular motion that it carries this fatty acid through over and over and over. And this process, once we've gotten to this point, will continue. So now instead of starting with an acetyl-ACP, we have a 4-carbon ACP. And we're going to add to it another malonyl-ACP. Every time we add something, we add a new malonyl-ACP. And every time we lose a carbon dioxide. And every time we go through this cycle, we add 2 more carbons. And the end product of fatty acid synthase action, is palmitic acid. Palmitic acid has 16 carbons. Yes, you should know that because palmitic acid is the most abundant fatty acid that you have in your body. Palmitic acid is a saturated fatty acid. This process makes saturated fatty acids. Palmitic acid? Yeah, Palmitic. You can call it palmitate, Palmitate. Okay? It's usually, as you may recall, linked to a CoA in the cell. Why? Because fatty acids are dangerous, right? So it's actually usually found in the cell as palmitoyl-CoA. But anyway, I accept any of those as being okay. We've just made a fatty acid. Now, let me review what I just told you. We start with an acetyl-CoA. Acetyl-CoA gets carboxylated by the enzyme acetyl-CoA carboxylase. That makes malonyl-CoA. Both acetyl-CoA and malonyl-CoA get converted to ACP versions. Now, we've got plenty of acetyl-CoA floating around here, right? They start the process. Malonyl adds to the adds to the acetyl-CoA, loses the carboxyl group, we've got a 4-carbon intermediate. Then we run them backwards of that process. And by the way, I should point out, there's a reduction there. There's another reduction down here. In both cases, the electrons are coming from NADPH. The overall process of all the stuff on the right is happening inside the enzyme known as fatty acid synthase, and this continues through several cycles. To make palmitic acid, it actually goes through 7 cycles, 7 times around and around. And the product of that is a 16-carbon intermediate known as palmitate. The good news, and there's more good news, is making the other fatty acids from palmitate is actually quite simple, at least from our perspective. How do we make unsaturated fatty acids? We use an enzyme called a desaturase. That's simple enough. Desaturases make fatty acids that have cis double bonds in them. But there's a catch. The catch is, in our cells, we cannot make double bonds farther than position 9 away from the carboxyl group. We can't make it beyond what's called delta 9. Yet we need fatty acids that have double bonds at position 12, position 15. How do we get those? We have to get those in our diet. So if we need things that have more double bonds out there than farther than position 9, we have to have them in our diet. Those fatty acids are known as essential fatty acids. Something we have to have in our diet is essential. We can't synthesize fatty acids that have double bonds farther than position 9. Okay? What if we want to make something longer than 16? Well, this is also simple from our perspective. We use an enzyme called an elongase. Okay, I'm keeping it simple by just giving it a name. Right? An elongase will synthesize longer fatty acids. Both elongases and desaturases are found in the endoplasmic reticulum. Consequently, we can make the things that we need. And if we can't make the things that we need, we have to get them in our diet. What's that? So the question is where they both found. So the desaturases and the elongases are both found in the endoplasmic reticulum. Okay. I just show you this. You don't need to know this, but this is what coenzyme A looks like. Okay. This is what ACP looks like. All right. Up to this point, they're identical on the left. All right. So in other words, when we think about coenzyme A, I said it was a handle that the cell can hang on to. Same handle. It's just attached to a different thing. In this case, it's attached to a protein. In this case, it's attached to ADP. All right. You guys are sounding tired. I'm going to finish with a song to summarize everything. Okay? And it's the same tune as yesterday, so it ought to be even easier for you. It goes as follows. The 16-carbon fatty acid palmitate gets all the carbons that it needs from acetate, which citric acid helps release from mitochondria matrices. Oh, a shuttle's great when acids are synthesized. Carboxylase takes substrate and it puts within dioxycarbon carried on a biotin. CoAs all gain a quick release replaced by larger ACPs, and it all begins when acids are synthesized. A malonate contributes to the growing chain. 2 carbons 7 times around again. For saturated acylates, there's lots of NADPH that you must obtain when acids are synthesized. Palmitic acid made this way all gets released. Desaturases act to make omega-3s. The finished products, big and small, form esters with a glycerol, so you get obese when acids are synthesized. All right, guys, have fun on the exam on Friday. I will see